Can you guys hear me?
Hello.
Hey, Olivia, how are you?
Pretty good. How are you? It's me and Blair here. We're in a big room and working on stuff.
It looks like you're in the PLC.
We are in the PLC. Yep.
Well, how's it all going?
It's going. Just kind of what we're doing now is kind of writing out like on the study guide, like on the whiteboard about like what we think about the main topics and just kind of working through them before. I'm just kind of figuring out what we aren't comfortable with, what we are comfortable with currently.
Uh-huh.
Can you hear Blair?
Hello.
I can't see Blair, but I can hear him.
Okay. I think really well walking around, so
No, that's fine. That's good. I'm sitting in my living room having a rum and Coke.
Okay, sounds
What
good.
kind of rum?
Oh, Bacardi.
Oh. I like it.
I just got back from a bike ride and I'm just chilling.
How far did you ride today?
21 miles or something like that. That's the normal Wednesday route.
Gotcha.
Hey, guys.
Hello.
Yeah, 20 and a half. All right. You guys ask me questions. I hopefully provide you answers.
So I not the best way to do this would just to be kind of just go down the list, make sure we know kind of what is up with this study guide.
Okay. I mean,
That sound good?
that study guide covers like 3 weeks of work, so we're not gonna get through it all in detail in an hour or two.
Yeah.
All right, so just be focused.
Okay. One thing that we didn't know is the specifics of the second one, the explain how genes are inserted into the genome of the Agrobacterium tumefaciens.
Okay. All right, I'm gonna share my screen with you.
Okay, awesome.
Let me pull. I pulled up all of the PowerPoints before I got on. So let me. So it's in the PowerPoint on genetic engineering, and it's towards the beginning of that PowerPoint. So can you guys see the gene modification?
Yeah.
Okay. All right. So after I had gone through and just kind of introduced the idea, we talked about different times in the history and stuff like that. So here we are. Well, I don't know what slide we're on, but this is Agrobacterium tumefaciens, and so this is a bacterium that causes tumors in plant cells. And the way it causes tumors in plant cells is that it inserts a part of its genome into the plant cell. And so what clever scientists basically did was they said, well, this plasmid, so there's a big bacterial chromosome, and then there's a little tiny plasmid. It's usually only, 1,000 or so base pairs. Yeah, you had a question?
When the Agrobacterium goes into the cell, they send in a complementary base strand?
No, so
is that correct?
Yeah. So naturally what they do is they break this plasmid open and they send it out of the bacterium and into the plant cell, and then it incorporates itself into the plant genome. But what scientists did was they cut it open themselves and put their own gene in. So they can use these things called restriction enzymes to cut the plasmid at a particular place, and then they can cut the foreign DNA at similar places so that they leave these sticky ends open that then can complementary base pair with the open part of the plasmid.
And that was that animation that we watched.
No, that was actually CRISPR, but it's a similar kind of idea because it's all based on complementary base pairing.
Yeah.
But in this case, it's just going to usually be 3 nucleotides that are hanging over here at the end, as opposed to in CRISPR, it's a really long segment of RNA that's complementary base pairing. So you're just using this as scissors, this enzyme as a pair of scissors to open up that plasmid. Then you stick this foreign DNA into that slot. So you see there are only four nucleotides hanging loose there. This is what's called a sticky end. And this thymine, thymine, adenine, adenine matches up with that adenine, adenine, thymine, thymine, and it just inserts itself in there. Now, when that bacterium goes to infect a plant cell, it's infecting the plant cell not only with its own genome, but also this new little section of DNA that scientists put in there. And then that then becomes incorporated into the plant's genome. And then once it's in there, the plant will just replicate that DNA just like it would replicate its own DNA, because it's actually a part of its own DNA now. So anytime it goes through the process of cell division, it's going to carry that gene along. Does that make sense?
Yeah.
Okay. So this is it in stages. So in the slide presentation, it's broken up in stages because I talked about it as we went. But then the last slide in the sequence is the whole thing from top to bottom.
Okay. All right. Anybody else have any questions about the study guide while I'm looking?
There, your nose is showing.
Oh, sorry.
They'll be giving Olivia the COVID.
I also had a question on like the Roundup Ready crops. What do
Yeah.
you mean by that?
Okay.
Oh.
So, I might have said it in passing, but I'm pretty sure I said it. Going back to, oh crud, did I close that? Or is it behind you guys? Doggone it. All right. I'm now on a single screen at home instead of 2 screens at school, which is where I usually am. So I'm having to navigate around a little differently. So can you see the genetic engineering slides again?
Mhm.
Yeah. So when we talk about which one should I use? Oh, so when we look at this, for example, we see, for example, we see GMO versus non-GMO. So genetically engineered versus non-engineered. And then when we look over here in this panel, we see that there's herbicide tolerant and insect resistant and both. Well, that herbicide tolerant is almost in all cases resistance to or tolerance of the herbicide glyphosate. So glyphosate, that's spelled Glyphosate. I think I wrote it on my whiteboard the day that we went through it. This glyphosate is the active ingredient that is in the commercially available herbicide Roundup. And so Roundup is used widely. If you see some dude spraying it on the grass coming up through the cracks in his sidewalk, chances are that's Roundup. You can go to Home Depot, you can buy it off the shelf. It's made by Monsanto. They have a patent on Roundup, and they also have the patent on the gene that makes plants resistant to Roundup. And so this class of crops that have been engineered such that a farmer can just spray glyphosate on his crops, his or her crops, and it will kill all of the weeds, but it will leave the corn and the soybeans alone. Those are called Roundup Ready crops. And it just means that they're herbicide resistant and they're resistant to the herbicide glyphosate because that herbicide is called Roundup. Does that help?
Yeah.
I'm pretty sure I didn't go into it in that level of detail, but I did refer to it as, I'm pretty sure I referred to it as Roundup Ready.
And never mind. Forgot I said anything. One more kind of in this area is kind of two more, I guess. One is the point that says what kind of traits were genetically engineered in common crop plants. Is that like pesticides, larger yields, and sturdier plants? Is it like what they look for?
No, so it's like, in this case, we've got herbicide resistance, HT, that's what HT is. We've got BT, which is the insect resistant, that has Bacillus thuringiensis genes in it. And then we have things like drought resistance. So if we go back a couple of slides, we have herbicide resistant. The flavor saver tomato was a gene that delays ripening so that it can be picked when it's green and shipped, and it doesn't ripen while it's in shipment. So the major classes, they're all in these slides, so you can see the flavor saver tomato. That's a delayed ripening. BT is an insecticide. Herbicide resistant tobacco, that's an herbicide resistant, a Roundup Ready crop. Let's see, herbicide resistant wheat, herbicide resistant. Yeah, so those are the big ones, and they're all in those couple of slides in the history portion of the genetic engineering talk. Does that help?
All right. Yes.
Okay.
That answered my second question too about BT, because our our chapter in the science report also talks a lot about BT, so I just wanted to make sure I knew what it was. Like,
Yeah,
for sure.
it's shorthand for Bacillus thuringiensis, and that's the species of soil bacterium that they got that gene from that kills insects.
Naturally occurring insecticide.
Yep.
That's common in soils, correct?
What's
Yeah,
that,
the where did
Olivia?
it come from?
I said it's common in soils, but like what we were confused about is where it came from. Is it just naturally occurring, or like I guess I just want to know.
Is that the answer to where does it come from?
Yeah, it's just naturally occurring in soil. It's a soil bacterium.
Awesome.
Yep.
I just had a quick question about the very last question. The polyphase farms, was that the same stuff we went over this morning, like with the rotation of like the cows and the chickens?
Yep, sure
Is
was.
that's what that is? Okay. I
Yep.
just wanted to make sure I had that right.
Mm-hmm.
Oh, hexaploid.
What's that, Blair?
Hexaploid.
Oh, hexaploid. Okay. So that's in the part dealing with
Yeah, no idea.
dealing with the history of domesticated plants. So I'm going to go to the evolutionary history of plant crops PowerPoint and share my screen. So here we are, evolutionary history of plants. So if we go down, we were talking about where different plants came from, and of course wheat comes from the Fertile Crescent region of the Middle East originally. And when we were talking about how wheat came into being, this is slide 12 of this day's material. And what you see here is that way back in human history, you basically had wild, what they call einkorn wheat, which is a type of wheat. And these are all diploid wheats. And when they cross this diploid wheat with this diploid wheat, rather than through the process of meiosis, them reducing the number of chromosomes, they actually ended up with both sets of chromosomes in the new species that was made out of this hybridization. And so that then became a diploid, it went from being a diploid wheat species to being a tetraploid wheat species. So you've got both copies of the A gene and both copies of the B gene. So you took this diploid wheat species, Triticum circe and Triticum monococcum, and when they crossed those, rather than getting a new diploid species, they got a tetraploid species, having four. It's instead of a 2N species, it's a 4N species. So it's got 4 copies of each chromosome. And then after a while of raising that wheat and cultivating that wheat species, they then crossed it with Triticum tauschae. And when they did that, once again, the genome of tauschae didn't get reduced as well. And so you went from a tetraploid wheat species to a hexaploid wheat species. So you've got six versions of the same chromosome, as opposed to us, who only have 2 versions of each of our chromosomes. So let me go back and pull something else up, just so you remember all of this and can put it in the right context. Let me go back to mitosis and meiosis, that PowerPoint, because I want you to understand the ploidy thing. So this should be the mitosis meiosis slides that you see now, correct?
Yes.
No? Yeah, okay. So if we are looking at our karyotype, so we have 46 chromosomes total. But what we have is we have 2 copies of 23 different types of chromosomes. We have 2 copies of chromosome 1, 2 copies of chromosome 2, 2 copies of chromosome 3, 2 copies of chromosome 4. Now, when we go through meiosis, this will be cut in half because you only get 1 copy of chromosome 1, 1 copy of chromosome 2, 1 copy of chromosome 3 in the gametes. So when that happens, you can have the gamete from one parent and the gamete from another parent come together, and then you have a diploid offspring out of 2 diploid parents. But what happens sometimes, and this happened in a frog species actually, is sometimes meiosis goes about where it doesn't actually divide the homologous chromosomes. And so in the gametes, rather than only having 1 copy of chromosome 1, you actually get 2 copies of chromosome 1. And that's what happens pretty commonly in plant species. And so oftentimes when you hybridize plants, you get the entire genome rather than just half the genome. And so you take a diploid parent species, another diploid parent species, you cross them, and rather than getting a diploid offspring species, you get a tetraploid. And so that's what happened in this case with wheat. So I'm going to go back to that. So what you have is diploid and diploid producing a tetraploid. This tetraploid then is crossed with a diploid, and that cross gives you a hexaploid. So 2 copies of 1 chromosome, you have now 4 copies of 1 chromosome, and then down here you have 6 copies of 1 chromosome. So ploidy deals with how many copies of the chromosomes you have, and hexaploid means that you have 6 of them. And so whenever you eat bread wheat, you're eating this bastardization of genetic modification just through crossing species, because you have this thing that doesn't really exist out in nature because it has way too many copies of the chromosomes. And that's your bread. Does that help?
Yeah.
Yes.
Oh my God, I'm giant on those screens.
I feel really weird walking around and then like looking at the camera and just seeing you wandering the room.
All right, what else you got?
So does anybody else have anything? I don't want to dominate the conversation if people want certain questions answered.
I don't have anything right now, so go for it.
Jared Diamond.
Yeah.
We have these 2 big paragraphs right here, and I remember us talking about this. How deep should we dive into this? Should we just like kind of know what's going on with the arguments that he makes, or should we go like responsible self-level critical thinking?
Well, you should certainly be familiar with his argument. So the first argument that I talked about was without agriculture, you would not have had civilizations. So
Yeah,
agriculture, what's that?
It's anybody in the Fertile Crescent would have become the dominating.
Yeah, so of all the agriculture, so you can't get societies arising, big complex societies arising without agriculture. And it just so happens that the Fertile Crescent had all of well, didn't have all of them, but had the most of any region in the world of all of these large grained plants and these large domesticable animals. And so in the Fertile Crescent, you had agriculture arising really early, a couple of thousand years earlier than agriculture took hold in China. You had the rise of city-states, you had the rise of governments, you had the rise of division of labor, you had development of writing systems, mathematics, et cetera, et cetera. And his argument is you couldn't have had any of that without agriculture, because agriculture allowed you to have a surplus of food, The surplus of food allowed you to have a lower death rate. Lower death rate led to larger population size. Larger population size needed more food, but because you were doing agriculture, you had a surplus of food, and so not everybody had to grow food. So other people could do other things like become mathematicians and learn about metallurgy and make bronze and steel and go and kill people. And because you were all packed together in a city now with a bunch of agricultural land around you, you also passed diseases from one another really easily. And so you also, over time, evolved a supercharged immune system because you were living around your filthy neighbors all the time. And so the presence of really successful agriculture led to large societies that were sedentary, that led to the development of technology, led to the development of disease resistance. And those are the things that then enabled those people to go out and kind of conquer the world.
Whereas
Guns,
these
germs, steel.
are
Go ahead.
just these hunter-gatherer societies on the opposite end are just food centric.
Yeah, they're just every day they're worried about getting food.
Yeah.
And the Aztecs and the Incas and the people in Papua New Guinea all might have gone down this path as well. But Diamond's argument is that in all of those cases, even though they got pretty big societies for their particular time in history, they all lacked something that made the Fertile Crescent and China for a while really excel and take off. So for example, the Incans, they had good agriculture. They had potatoes. They had llamas, but they didn't have any other large mammals that could easily be domesticated. And even if you hung around llamas very often, they're not the greatest domesticated animal in the world. They have a little bit of a temper even. The next biggest dietary, the next biggest meat bearing animal that they have in the Andes are guinea pigs. So you've got a llama, which is not a terribly big animal, and then the next step down for you is a guinea pig. And I've been to the Andes, I've been to Peru, and I've had guinea pig. And let me just tell you, it takes a number of guinea pigs to fill you up. And his argument is you can't really build a Fertile Crescent-sized complex society on llamas, potatoes, and guinea pigs. Is kind of the thrust of his conversation. In Central America, they had complex societies, but once again, while they had a good suite of crops, beans and squash and corn, they didn't have any large domesticable animal. Their biggest animal that they ate on a regular basis were turkeys. So once again, hard to build a really big complex society capable of building ships and going abroad and conquering the world on corn, beans, squash, and turkeys is kind of the point. And there are all kinds of things that you can criticize in there, and he's taken kind of a beating from historians taking this ecological and evolutionary approach to things, but you know, that's that. So then the other part, so that's the first part of, I think, the two diamond phases. I don't have the study guide in front of me.
The second one is it's two things, which the second thing is the one I have a question about. The first one is the 6 characteristics, which I know are on the PowerPoint. So we're just supposed to know that list.
What's that?
We're just supposed to know that list.
Yeah, so in order to successfully domesticate a large mammal, it's good if it's an herbivore because that's energy efficient. It's good if it has a social hierarchy. It's good if it has a disposition that makes it not want to kill you. It has, it grows relatively quickly, so you don't have to wait forever for it to grow up.
Yeah.
Why am I blanking on a couple of other ones?
Is one of them that's not nomadic? That it stays?
Oh yeah, it occurs in herds.
Yeah. Because the water buffalo have that.
Oh yeah.
Yeah. So,
why am I forgetting the 6th one?
So it's social hierarchy, grow quickly, They occur in herds. They won't kill you. I missed the first one you said.
What was the first one that I said?
You said
Oh,
one,
they're
and then you
herbivores.
said
Herbivores.
herbivores. Herbivores.
Because if you're looking at carnivores, it takes just tons of energy because you have to raise herbivores to then feed the carnivores. And so we'll study this after the exam, we'll study energy transfers in food webs.
Yeah.
Oh, and you have to be able to breed it in captivity. That was the one I was forgetting. Probably because it's so obvious. So for example, rhinoceri are very large animals, but rhinoceri, they are herbivores, but they don't grow terribly fast. They're really hard to breed in captivity. They have kind of a rough disposition, and they're solitary, except when they're going to mate.
What was the two things that Fritz Haber did? That's where I have the question.
The two things what?
That Fritz Haber did?
Fritz Haber. This is a different thing altogether. Winston, what did Fritz Haber do?
He was responsible for the industrialization of nitrogen fixation.
Yeah,
And
so
then
he
didn't
invented
he get a? Oh, go ahead.
Yeah, so he invented industrial fixation of nitrogen. Yep. The process for it, the Haber-Bosch process. What else did he do? Mary?
He won the Nobel Prize for it, and
He did?
they used the nitrogen fixation to like create like deadly gases in World War II.
No, you're confusing some things. They used
Okay.
the fixation of nitrogen for fertilizer, which was kind of why he developed it.
Uh-huh.
But ammonium nitrate, which is the fertilizer, is also used in making gunpowder. So ammonium nitrate is good for putting on your fields, but it also enables Timothy McVeigh to blow up the federal building in Oklahoma City. The bomb that Timothy McVeigh used to blow up the federal building, the Murrah Federal Building in Oklahoma City, was a fertilizer bomb. He basically got some fertilizer, ammonium nitrate, from his uncle's or cousin's farm up in Michigan. It was a soybean farm, actually, and mixed it with fuel oil and created what they call a fertilizer bomb. And so ammonium nitrate is a key component of munitions. So those two things are tied to one another. But the other thing that he did, in addition to industrial fixation of nitrogen, is he also worked on developing pesticides. Once again, because he was a chemist who was interested in agriculture. And as it turns out, some of these pesticides ended up being used by Germany during World War I, and then were used by Germany again in World War II. The most famous of his pesticides was Zyklon B, which was used to gas Jews in the gas chambers at Auschwitz, type of cyanide containing compound. So the thing about pesticides is they kill insects just fine, but they also kill humans. So you can turn those compounds to, the benefit of human beings or the detriment of human beings, depending on how depraved you are, I guess is one way of saying it. So both of his kinds of inventions, both of his development of pesticides and development of ammonium containing compounds that are used as fertilizers, both of those things had upsides and downsides. Great increases in agricultural productivity because of fertilization, and increases in agricultural productivity because you got rid of pests, but then those things were also subverted by evil people. Because humans are humans.
Nitrogen limiting is because of leaching, right?
Nitrogen is limiting for two reasons. One reason is because of leaching, and the other reason is because there's just a limited number of organisms who can get nitrogen out of the air and into forms that are useful for organisms. And those are mainly soil bacteria, cyanobacteria, and bacteria found associated with the roots of legumes. So two reasons: hard to get, and it gets lost through things like leaching. And denitrification. Taking that useful nitrogen that is in the soil, bacteria break it down, get energy from it, and blow it back to the atmosphere as nitrogen, diatomic nitrogen, gaseous nitrogen. So what else you got?
With the biological and macromolecules, will we just have to like identify those, or do we need to know anything else about them?
So you need to, so there's actually a question on the exam 2 that I posted. And in that question, it has a list of the different kinds of compounds. So carbohydrates, lipids, nucleic acids, and proteins. And then it has a list of the elements and it asks you to circle which elements are found in which one. So that's one kind of question that I could ask you. The other kind of question that I could ask you is I could just show you some simple images similar to the images that are in the PowerPoint that show you, here's a chemical compound. Is this a protein? Is this a lipid? Is this a carbohydrate? Is this a nucleic acid? And once again, when I went through it in class, I said, one of the things that you might want to do is figure out some key indicators for which ones of these things they might be. So carbohydrates are almost always in nature found in ring forms. And they're ring forms that have a lot of carbons around them and a lot of hydrogens, but they're ring forms and they have a bunch of OH groups hanging off of them. So sugars will be easy to recognize in that way. Lipids, on the other hand, they have a carboxyl group and then they have these long chains of carbons, usually with a lot of hydrogens around them. And so whenever you see long chains of carbon with lots of hydrogens around them, that's more than likely going to be a lipid, especially if it's attached to the glycerol molecule, which is a bunch of carboxyl groups all put together. Carbons double bonded to oxygens and single bonded to another oxygen. Nucleic acids are easy because they all are chains that are sugar phosphate chains with nitrogenous bases attached to them. And then amino acids are just repeating units proteins are just repeating units of amino acids. And the amino acids have a particular structure to them. They have an amino group on one side, an NH2 group on one side. They have a carboxyl group on another side. They have a central carbon, and then the side chain off of that varies. And so, once again, just kind of, and I guess I could have shown these to you rather than just waving my hands in the air while I did that. So let me go and do that actually. I pulled these PowerPoints up so I would have them available to me. So can you see this?
Yeah.
So here's the carbohydrates. They're these ring forms with carbons and hydroxyl groups, OH groups attached to them. Lipids have a glycerol molecule, a carboxyl group, and then these long chains of carbons, typically with a lot of hydrogens, but occasionally the hydrogens are replaced with double bonds. Sometimes these glycerols, the glycerol is replaced by a phosphate in the phospholipids, which are used in cell membranes. Amino acids, which are the building blocks of proteins, have a carboxyl group on one side, an NH2 group on the other side, a central carbon with a side chain, and the side chain varies, and these side chains vary quite a lot. So once again, here you have a carboxyl group, here you have the amine group, and then here you have the side chain. Carboxyl, amine group, side chain. Carboxyl, amine group, side chain. And then nucleic acids always have a phosphate, sugar, phosphate, sugar, phosphate, sugar backbone with these nitrogenous bases. You know that they're nitrogenous bases because they have tons of nitrogen in them, these ring structures. This is DNA because it's a double-stranded molecule. This is RNA because it's a single-stranded molecule, and it has uracil as one of its nitrogenous bases rather than thymine. So once you kind of know what the hallmarks of them are, they're not that difficult to tell apart. Does that make sense?
Mm-hmm.
Okay. And I wouldn't try to learn all the details. I would just learn the big things that make them different.
Okay.
If you want to know all the details, go and take a biochemistry class. But don't. It's hard and boring. Don't do it. I took biochemistry as an undergraduate. I hated it.
Various flows of nitrogen in the nitrogen cycle. We just have to know the beginning and end products.
Mm-hmm.
That's in a PowerPoint.
And chances are, if I'm going to ask you about nitrogen cycling stuff, I'm probably going to give you the nitrogen cycle. And so one of the nice things about having that nitrogen cycle in front of you is that you can figure out what the beginning and end products are if you pay attention to the details of the cycle.
You'll give us that like image of the.
Yep. So like I told you earlier today, what I typically do is I give you the image and then I ask you a series of questions about the cycle, rather than ask you to produce this cycle and then ask you a bunch of questions on it, because I don't really expect you to be able to do that. And it's not really necessary for a class like this. If you're taking an upper level ecology class, I might ask you to do that, but I might not even ask them to do that. But yeah, you should be familiar with the fluxes and be familiar with the beginning and end products of the fluxes. And I would make the table kind of like I was starting to make in class today. What's the flux? What's the beginning? What's the end? Where does it occur? Who does it? Is not a bad way to study that.
Okay. If people want to go over it, we can, but it's a quick thing. The carbon cycle imbalance is because more carbon in the air means more infrared in the atmosphere.
No, it's unbalanced because there's more carbon dioxide going back to the atmosphere than is being removed from the atmosphere by plants. And so without humans, it would be unbalanced because there would be more carbon dioxide coming out of the atmosphere into the terrestrial ecosystem than is being returned. But now it's imbalanced in the opposite direction because there's more carbon dioxide going into the atmosphere than is being stored in ecosystems. So in both ways, it's imbalanced, but one is an imbalance that stores carbon in ecosystems. The other imbalance returns carbon to the atmosphere.
Okay.
Does that make sense?
Humans just cannot catch a break.
So the second part of what you said, as this carbon dioxide builds up in the atmosphere, it does create global warming. That's not the imbalance. The imbalance is the flow. Where is it flowing? Where is it being stored? What's that?
That's kind of the end result of the imbalance. Is that?
Yeah. So the implication of that imbalance is global warming, but the global warming isn't the imbalance.
That's what the polyphase forms into. I think the only thing we really haven't talked about is about sickle cell and all the things associated with that.
So what is sickle cell? What is sickle cell anemia?
Yeah, I'm just looking. There's like a couple of things on it. I think that's the
Okay.
only thing in the study guide, really. So it was. Where is it? There we go. So the sickle cell, going back, is what they use CRISPR on.
Yeah, so step back a minute and ask yourself, what is sickle cell anemia? What is it, and what kind of disease is it?
Isn't it a major deficiency of iron?
It is not a major deficiency of iron. It's a mutation. It's a single nucleotide polymorphism that occurs in the hemoglobin gene that causes the structure of hemoglobin to be altered such that rather than being a nice donut-shaped cell, it forms a sickle, and it doesn't carry oxygen as well as a result of it. But it's just a single nucleotide in there are 4 different proteins in hemoglobin, and it's just a single nucleotide in one of those 4 proteins that makes this difference. So it's a genetically inherited disease and one single nucleotide polymorphism. So all of the nitrogenous bases and all of the DNA that make up all 4 of those proteins, there's just one change, one different nitrogenous base that changes one codon, that changes one amino acid, and totally messes things up. Now, the upside of sickle cell anemia is it makes you more immune to malaria. But when you're not being afflicted by malaria, it messes you up in a lot of other ways. And it's evidently a very painful chronic illness that certain subpopulations of humans have because they evolved in places where there was malaria. So it's great when you have malaria. It sucks when you don't. But it's just one nucleotide that is different.
So, in terms of CRISPR, it's used for genetic, or for hereditary diseases.
Yeah, so they took an adult woman and they CRISPR'd her. They killed off, what's that?
How much will we have to know about the woman with sickle cell?
So you need to know enough because I spent some time on it in class. So you have cells in your body that make new blood cells. So they harvested those blood cells, some of those stem cells that make new blood. They harvested them from her. They CRISPR'd them. But then before they could put those cells back in her, they had to kill off all of her other cells that make blood. Otherwise, there would be too many of those things making sickle cell anemia blood in order for the CRISPR blood cells to kind of overcome that. So they took some of her stem cells out, CRISPR'd them, killed off all of her other stem cells, and then put the CRISPR'd stem cells back in. Those CRISPR'd stem cells make normal hemoglobin, and a year into it, it kind of seems like she's cured, which is fairly amazing.
Right.
But the nice thing about it is that the change was a really tiny genetic change, and it had to be done at exactly the right place, and that is exactly what CRISPR is really good at doing.
Yeah, that answered my question. I just want to know, like, did we have to know, like, what the doctors did, or like, do we have to know, like, about her specific case?
Well, at the level that I just gave it to you, that's kind of the level you need to know it at.
Yeah.
Yep.
Yeah. Okay. Awesome. Yeah. Does anybody else have any other questions?
That's all I had.
Yeah, that's good.
Man, if that's all it took, why did we spend 3.5 weeks on this stuff?
Domesticated corn.
Domesticated corn, yeah.
Yeah. The phenotypic changes that occurred. Hold on, we're starting another bullet list. If I remember correctly, and that's not a guarantee. They were This was in the Poland book, right?
It wasn't really. It was mainly in stuff that I gave you in class. Let me go and find that.
Sure.
Where is that?
I remember him talking about corn adapted to where it almost had to be farmed by humans in order to survive,
Oh.
and that's how it kind of.
Yeah, so that's part of it, because the ears are sheathed in leaves now, and that was not the case in the past. And I think I'm mistakenly closed that. Let me go and get it. So corn. Okay. So here's corn. The Zia is corn in this evolutionary tree. You see that it is related to sorghum, and you also see that it is related to wheat and rice. There are all these grain crops. And so this is teosinte, so this is ancient corn. This is the wild progenitor of corn. And what you see is that teosinte is very small. It has a reduced number of kernels on the stalk. And so among the changes that have occurred in corn is the kernel count has gone up. The length of the ear has gone up, has gotten bigger and bigger. The diameter of the ear of corn has gone up. And if you go back even before this, where was it? Even the structure of the plant changed, and I'm trying to find which presentation it is in. Oh yeah, it's in the genetic engineering presentation. So it's slide 4 of the presentation on genetic engineering. And let me show you that image as well. So here you see two things. You see, can you see the corn? You can see the change in the morphology of the ear from little tiny ears with relatively few kernels to longer and thicker ears with lots more kernels. But then when you look at the teosinte plant, you see the teosinte plant is a multi-stalked plant. And if you blow this up a little bit, you can see that it has lots of little ears all along the stalks. So each branch has multiple ears on it. The flowers are still separated. You still have male flowers up here and female flowers down here. But over time, the corn plant itself has undergone a change such that it's just a single stalk. And along this single stalk, there are usually just 1 or 2 ears of corn. So over time, the plant has gone from lots of stalks with lots of little ears to a single stalk with a few large ears. And so those are kind of the major morphological changes. And those changes are all related to a relatively few number of genes. It only took about changes in 7 genes to transform teosinte into what is now modern corn, which is pretty impressive because that change was wrought by a bunch of indigenous people with very little knowledge of biology and no knowledge of genetics. And that's mentioned in the notes. If you go down to the notes underneath the, I don't know if you can see it on my screen, but it talks about this down in the notes section of this slide. 7 genes of large effect.
Why do organisms need nitrogen, phosphorus, sulfur, and carbon?
Why do organisms need nitrogen, phosphorus, carbon, sulfur, all those things? Mary, you asked a question about this earlier.
Isn't it just because that's like the building blocks of pretty much everything, like the biological macromolecules?
So if you guys go back to the nutrient cycling PowerPoint slides, which are here before you, why do organisms need carbon? Well, carbon is a major component of both glucose as well as carbohydrates rather, as well as a super huge component of lipids. Less of a component, but still something of a component to amino acids. But amino acids have a lot of nitrogen in them because they have the amino group. And also amino acids have nitrogen sometimes as parts of these side groups. They also have sulfur as some of these side groups as well. And you need phosphorus because of the phospholipids that are used in membranes. That is a type of lipid. And you need phosphorus in the phosphate sugar backbone of DNA. And you need nitrogen because of the nitrogenous bases of DNA. So you need all of these things because these things go into the major biological macromolecules that make up everything that is alive. Phosphate is also used in ATP, which is how you move energy around inside your body easily. Adenosine triphosphate has 3 phosphate molecules bonded to adenosine, which is a type of nitrogenous base, or a type of nucleotide rather, which has a nitrogenous base on it. So, yeah, they're all in you. If you didn't get nitrogen, you would be dead. If you didn't have a source of phosphorus, you would be dead.
No, I think that covers. I feel good.
Okay. Well, I'll be around tomorrow as well. I have office hours from 9:30 to 10:30, 9:35 to 10:35, something like that. So I'll be around in the morning. I'll probably be on campus also for a while because I'll be working on an exam. So if you have a question, just drop by my office.
Alrighty.
All right. You guys take care. Don't stay up too late.
Okay.
Thank you.
All right. Sure, no problem.